Picture this. A doctor sits across from a patient in a room that was specifically designed to feel calm and probably doesn't. There are pamphlets on a small table that nobody is reading. The doctor has delivered this kind of news before, more times than any human being should have to, and they're doing it with the practiced, careful language that training and experience produce. The patient is hearing words that rearrange the architecture of their entire future in real time. The word cancer has just been said out loud. And somewhere in the back of both of their minds, behind the immediate logistics of next steps, referrals, treatment timelines, sits a question that the patient is too shocked to ask, and the doctor has heard 100 times and still doesn't have a fully satisfying answer to why, after decades of research and billions of dollars and some of the most brilliant scientific minds in human history pointing themselves at this problem, is cancer still so devastatingly difficult to cure? We have mRNA vaccines that were developed in under a year. We have mapped the entire human genome. We have surgical robots, targeted radiation delivery systems, drugs that hit molecular targets with extraordinary precision. We have treatments that would have looked like science fiction to oncologists practicing 50 years ago, and cancer still kills approximately 10 million people per year globally. Still ends lives that should have continued, still defeats treatments that seemed to be working, still comes back. The answer to why is not simple, and it is not a story of insufficient effort or misallocated funding. It is a story about what cancer actually is, at the biological level, at the evolutionary level, at the cellular level, and why that reality makes it fundamentally different from almost every other medical problem humanity has ever tried to solve. The first and most important thing to understand about cancer is the thing that makes it categorically different from essentially every other disease that medicine has successfully addressed. Cancer is not foreign. It is not an invasion. It is not something that entered the body from outside, something that can be identified as alien and targeted as other. Cancer is you. Cancer is your own cells, running your own genetic code, using your own cellular machinery, expressing your own proteins, doing all of that in a way that has gone catastrophically wrong. The mutation that initiates a cancer cell's transformation came from your DNA. The proteins on the surface of that cancer cell are your proteins. The metabolic processes it uses are your metabolic processes, altered but derivative. When your immune system scans a cancer cell looking for something to destroy, it is often looking at something that appears in most of the ways the immune system uses to evaluate threat, like a legitimate member of the community. This is not a minor technical detail. It is the central obstacle. Every successful treatment in the history of infectious disease, antibiotics, antivirals, antifungals, vaccines, works by exploiting the difference between the pathogen and the host. Penicillin targets bacterial cell wall synthesis, a process that human cells don't perform. Antiviral drugs target viral replication machinery that has no equivalent in human cell biology. The drugs can be relatively selective because the target is genuinely foreign. With cancer, that distinction collapses. Any weapon precise enough to kill a cancer cell that is 98% biologically identical to a healthy cell carries the risk of killing the healthy cell too. This is why chemotherapy, the oldest and still widely used treatment approach, produces the side effects it does. Classic chemotherapy drugs target rapidly dividing cells. Cancer cells divide uncontrollably, which is part of what makes them dangerous, and targeting cell division seemed logical. But human bodies contain many populations of rapidly dividing healthy cells. The cells lining the digestive tract, hair follicle cells, bone marrow cells producing blood cells, and immune cells. When chemotherapy hits rapidly dividing cells without distinguishing cancer cells from healthy ones, it hits all of them. The nausea, the hair loss, the immune suppression. These are the consequences of attacking cell division in a system where cell division is also happening legitimately and critically everywhere. The second fundamental problem is that cancer is not one disease. This seems obvious when stated directly, but its implications are more radical than the statement implies. Lung cancer and breast cancer and leukemia and melanoma and pancreatic cancer are not variations on a single theme the way that, say, different strains of influenza are variations on a viral respiratory infection. They are fundamentally different diseases that happen to share the word cancer, because they all involve uncontrolled cellular proliferation. The underlying genetics, the cellular machinery involved, the growth factors driving proliferation, the interactions with the immune system, the response to treatment, the likelihood of spreading to specific other tissues, all of these differ not just between different cancer types, but between different patients with nominally the same cancer type. Two people both diagnosed with breast cancer may have tumors driven by completely different molecular mechanisms. One tumor is fueled by estrogen receptor signaling. Blocking that signaling with drugs like tamoxifen produces dramatic results. The other tumor lacks estrogen receptor expression entirely. The same drug does nothing. One tumor has a specific mutation in a gene called HER2 that makes it responsive to targeted antibody therapies. Another doesn't. The same diagnosis, the same anatomical location, the same histological appearance under a microscope, completely different molecular diseases requiring completely different treatment approaches. This is why the oncology research community increasingly speaks not of cancer types, but of cancer subtypes, and increasingly of individual patient tumor molecular profiles. The aspiration of precision oncology, sequencing the specific mutations in an individual patient's tumor, and selecting treatments targeting those specific alterations, represents an acknowledgement that the diversity of cancer is so profound that population-level treatment protocols have fundamental limitations. But the practical implementation of this aspiration runs into obstacles of its own. Tumor heterogeneity is the problem that keeps oncologists awake at night, and it represents perhaps the most devious aspect of cancer biology. A tumor is not a uniform population of identical cells. Even within a single tumor in a single patient, there is often dramatic genetic diversity. Different cells carrying different combinations of mutations, different subpopulations with different surface proteins, different metabolic dependencies, different sensitivities to treatment. This heterogeneity arises through the same evolutionary process that produces all biological diversity, just compressed into the growth of a tumor rather than the evolution of a species. As a tumor grows, mutations accumulate at random. Most are neutral or harmful to the cancer cell. Cells with those mutations die or don't proliferate efficiently. But some mutations confer advantages: faster growth, better ability to evade the immune system, resistance to a drug that's being used in treatment. Those cells are selected for, their descendants proliferate preferentially. Subpopulations arise with specific adaptive advantages. Treatment applies the most intense selection pressure of all. When chemotherapy or targeted therapy is administered, it kills the sensitive cells, often dramatically and quickly. Tumor burden drops, scans show response, the treatment appears to be working. But within the surviving population, there were cells with pre-existing resistance mutations, mutations that existed before treatment began, not mutations that developed in response to treatment. Those cells weren't responsive to the drug, so they weren't killed. While the sensitive cells were dying around them, the resistant cells had room to proliferate. Within weeks to months, the resistant subpopulation has expanded to become the dominant population. The cancer has relapsed, and now the treatment that worked before doesn't work anymore. This process, the evolutionary selection of resistance, is not a failure of the drug. The drug killed what it was supposed to kill. It is a failure of the treatment strategy, and understanding it requires thinking about cancer not as a disease to be eliminated, but as an evolving system that adapts to whatever pressure is applied to it. The cancer microenvironment adds another layer of complexity that research has only begun to map with adequate resolution in recent decades. A tumor is not simply cancer cells floating in normal tissue. It is a structured ecosystem. Cancer cells actively recruit and reshape the cells around them. Blood vessels grow into the tumor through a process called angiogenesis, supplying the oxygen and nutrients that fuel rapid growth. Fibroblasts, structural cells normally responsible for maintaining connective tissue, are co-opted into cancer-associated fibroblasts that provide structural support to the tumor and produce growth factors that stimulate cancer cell proliferation. Immune cells infiltrate the tumor, but often find themselves functionally suppressed rather than active. The tumor microenvironment actively converts the immune response from tumor attacking to tumor tolerating through a sophisticated array of molecular signals. The immune suppression within the tumor microenvironment is one of the most important problems in cancer biology because the immune system is in principle capable of recognizing and eliminating cancer cells. This happens all the time. Cancer cells arise constantly as a consequence of normal DNA replication errors and environmental mutagen exposure, and the immune system destroys most of them before any clinical tumor develops. The cancers that become clinically apparent are the ones that have evolved the ability to evade this immune surveillance. The evasion strategies are diverse and sophisticated. Cancer cells downregulate the surface molecules that allow immune cells to recognize them as abnormal. They secrete molecules that convert attacking immune cells into suppressive regulatory cells. They express surface proteins, particularly PDL1, which interacts with a receptor called PD1 on T cells that activate a braking system normally used to prevent autoimmune damage, essentially telling attacking immune cells to stand down. The tumor builds builds its own immunosuppressive bubble. This is what makes checkpoint inhibitor immunotherapy, one of the most significant treatment advances of the past decade, so important conceptually. Drugs blocking the PD-1/PDL1 interaction remove the molecular brake that tumors exploit to suppress immune attack. The immune system, relieved of the suppressive signal, can recognize and attack the cancer more effectively. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content, covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. In patients whose tumors are driven by high mutational burden and therefore present many abnormal proteins for the immune system to recognize, checkpoint inhibitors can produce dramatic, durable responses. Patients who would have survived months have survived years. Some appear to be cured, but checkpoint inhibitors don't work for all patients, don't work for all tumor types, and produce serious autoimmune side effects in a significant fraction of patients who do respond, because the same braking system the drugs disable was also protecting normal tissue from immune attack. The immune system, once released from the tumor's suppressive control, sometimes attacks the lungs, the colon, the pituitary gland. Balancing immune activation against autoimmune damage is a problem that doesn't have a clean solution. Metastasis is the process that transforms most cancers from local problems into systemic life-threatening diseases, and it is one of the most poorly understood aspects of cancer biology despite decades of focused research. Approximately 90% of cancer deaths result not from the primary tumor, but from metastatic disease, from cancer cells that have left the original tumor, traveled through the bloodstream or lymphatic system, and established secondary tumors in distant organs. The liver, the lungs, the brain, the bones are common metastatic destinations for many cancer types, and once cancer has spread to these sites, treatment becomes dramatically more difficult and prognosis dramatically worse. What makes metastasis so scientifically challenging is that it is extraordinarily inefficient, yet catastrophically consequential. Studies of cancer cell behavior suggest that millions of cells may enter the circulation from a growing tumor, yet only a vanishingly small fraction of those cells successfully establishes metastatic colonies. The cells must survive in the circulation, evade immune attack in the bloodstream, extravasate into a distant tissue, survive the completely different microenvironment of that tissue, and then either begin proliferating immediately or enter a dormant state that can persist for years before reactivating. The dormancy question is particularly clinically important. Some cancer cells can persist in distant organs in a non-proliferating dormant state for years or decades after apparently successful treatment of the primary tumor. They are invisible to imaging, unaffected by treatments targeting rapidly dividing cells, clinically silent. Then, triggered by factors that aren't fully understood, hormonal changes, immune system alterations, aging, infection, stress, they reactivate and begin proliferating in the distant organ. A patient who was declared disease-free 5, 10, 15 years after initial treatment develops metastatic disease that traces its origin to dormant cells that survived the original treatment. This is the biology behind the clinical phenomenon of late recurrence that patients who have been through cancer treatment and their families know, with dread, is always a possibility. The blood-brain barrier presents a specific version of the cancer treatment problem that illustrates how the body's own protective systems can work against treatment efficacy. The blood-brain barrier is a selective filtering system that tightly controls what can pass from the bloodstream into the brain, essential for protecting the brain from pathogens and toxins. For cancer treatment, this protection becomes an obstacle. Most chemotherapy drugs cannot cross the blood-brain barrier in therapeutically meaningful concentrations. This means that brain tumors, both those that originate in the brain, like glioblastoma, and metastatic tumors that have spread to the brain from elsewhere, are treated with a reduced pharmacological toolkit compared to tumors in more accessible locations. Glioblastoma, the most common and aggressive primary brain tumor in adults, has a median survival from diagnosis of approximately 15 months with current best treatment, surgery, radiation, and chemotherapy with a drug called temozolomide. That median has moved minimally in decades despite enormous research investment. The combination of the blood-brain barrier-limiting drug delivery, the tumor's infiltrative growth pattern making complete surgical removal impossible, and the extraordinary intratumoral heterogeneity of glioblastoma producing rapid resistance makes it one of the most resistant cancers to treatment advances. Hundreds of clinical trials for glioblastoma have failed to demonstrate demonstrate significant survival improvement beyond the current standard of care. The financial and logistical dimensions of cancer drug development contribute to the treatment gap in ways that are less biological but equally consequential for patients. Developing a new cancer drug from initial target identification to clinical approval takes on average over a decade and costs somewhere in the range of $1 to $2 billion per approved drug, accounting for the drugs that fail in development. The failure rate is extraordinary. The vast majority of compounds that show promise in laboratory and animal studies fail in human clinical trials, often because the tumor biology in a mouse model is too different from human tumor biology for mouse results to reliably predict human outcomes. Mice don't naturally develop cancer with the same genetic complexity and heterogeneity that human cancers display, and the mouse immune system's interaction with tumors differs from the human immune systems in ways that make immunotherapy results in mice particularly unreliable predictors of human outcomes. The translation problem, the gap between laboratory findings and clinical efficacy, has been a persistent obstacle in oncology. Compounds that cure cancer in mice with notable regularity have filled the research literature for decades, producing justified skepticism about mouse model results that has been hard to act on because mouse models are faster, cheaper, and more experimentally tractable than anything closer to human biology. Patient selection for clinical trials, identifying which patients are most likely to respond to a given treatment based on the molecular characteristics of their tumor, has improved significantly with better molecular profiling technology. But the fragmentation of cancer into many molecular subtypes means that clinical trials often struggle to enroll enough patients with the specific tumor profile being studied to reach statistical conclusions with confidence. Rare tumor subtypes, even if there is a clearly promising treatment, may have insufficient patient numbers globally to run definitive clinical trials through conventional pathways. The most promising directions in current cancer research represent genuine paradigm shifts rather than incremental improvements on existing approaches, and understanding them clarifies both why hope is warranted and why timelines for clinical impact remain uncertain. CAR T-cell therapy, chimeric antigen receptor T-cell therapy, involves removing T cells from a patient's blood, genetically engineering them to express a receptor specifically targeting a protein on the patient's cancer cells, growing millions of these engineered cells in the laboratory, and infusing them back into the patient. The engineered T cells then hunt down and kill cells displaying the target protein with remarkable efficiency. In certain blood cancers, particularly B-cell leukemias and lymphomas, CAR T therapy has produced complete remissions in patients who had exhausted all other options. Long-term follow-up data suggests that some of these responses are durable, potentially representing cures in patients who would previously have had no remaining treatment options. The challenges facing CAR T expansion into broader cancer application are substantial. Solid tumors, the majority of cancers, are much harder to treat with CAR T than blood cancers, partly because the engineered T cells have difficulty infiltrating the tumor microenvironment, and partly because solid tumors don't have the clean, uniform surface protein targets that make blood cancer targeting tractable. Identifying a surface protein expressed uniformly across all cells in a solid tumor, but absent from critical normal tissues, is extraordinarily difficult, given the heterogeneity that characterizes solid tumor biology. The manufacturing complexity and cost of CAR T therapy are also significant barriers. Current CAR T products require manufacturing processes that take weeks, cost hundreds of thousands of dollars per patient, and must be produced individually for each patient from their own cells. Allogeneic CAR T, engineered from donor cells rather than the patient's own, offers potential for off-the-shelf manufacturing, but introduces immune rejection challenges that haven't been fully resolved. Cancer vaccines represent another frontier that has been more difficult than early optimism suggested and more promising than recent pessimism implies. The mRNA vaccine technology that produced COVID-19 vaccines with remarkable speed is now being applied to personalized cancer vaccines. Vaccines tailored to the specific mutational profile of an individual patient's tumor. Tumor mutations produce abnormal proteins called neoantigens that are present on cancer cells but not on normal cells, making them ideal immune targets. Vaccines designed to train the immune system to attack cells displaying a patient's specific neoantigens could in principle produce immune responses precisely targeted to that patient's cancer. Early clinical results in melanoma and pancreatic cancer have been encouraging enough to drive large-scale clinical trials, and the technology platform developed for COVID vaccines makes the manufacturing of personalized personalized vaccines far more feasible than it would have been a decade ago. But personalized cancer vaccines require tumor sequencing, neoantigen prediction, vaccine manufacturing, and administration on a timeline that fits the clinical reality of the patient's disease, a logistical and biological challenge that the field is actively working to address. The philosophical reframe that some cancer researchers have embraced, treating cancer as an evolutionary process to be managed rather than a foreign agent to be eliminated, has produced strategies that depart meaningfully from the dominant treatment paradigm. The conventional approach to cancer treatment is to apply maximum force as early as possible, dose chemotherapy and targeted therapy at the highest levels patients can tolerate, with the goal of killing as many cancer cells as possible as quickly as possible. This approach makes intuitive sense and often produces dramatic initial responses, but it also applies the strongest possible selection pressure for resistance, and it is resistance that ultimately defeats most treatments. Adaptive therapy, a strategy developed from evolutionary game theory, takes the opposite approach. Rather than maximizing cancer cell kill, adaptive therapy aims to maintain a stable, manageable tumor burden that includes sensitive cells. The sensitive cells, in the presence of treatment, outcompete the resistant cells for space and resources. If treatment is titrated to maintain sensitive cell populations rather than eliminate them, the sensitive cells actively suppress resistant cell expansion. The tumor is controlled rather than eliminated, but it is controlled in a way that doesn't inadvertently select for the resistant population that eventually defeats elimination strategies. Clinical trials of adaptive therapy in prostate cancer have shown longer times to treatment failure compared to conventional dosing in some patient populations, providing proof of concept that the evolutionary framing can produce clinically meaningful improvements. The conceptual shift from war metaphor to ecological management is significant, though whether it will translate to better long-term outcomes across cancer types remains an active area of investigation. Return to that consulting room, the pamphlets nobody is reading, the doctor with practiced, careful language, the patient whose future just reorganized itself. What neither of them can say honestly is that there is a cure. What both of them know is that the treatments available are better than they were 10 years ago and substantially better than they were 20 years ago, and that the fundamental scientific understanding of what cancer is and how it operates has advanced in ways that are producing increasingly effective interventions even when the word cure remains out of reach for most diagnoses. The difficulty of curing cancer is not a failure of effort or investment or intelligence. It is a reflection of the fact that cancer is, at its core, evolution happening inside your body, your own cells accumulating mutations, the variants best adapted to their environment proliferating, treatments applying selection pressure that drives adaptive responses. Fighting evolution with drugs is different from fighting bacteria with drugs because bacteria are foreign and evolution is the process that built you. You can't easily target a process that is you. What you can do is understand it with increasing precision. Develop treatments that target its specific molecular drivers. Harness the immune system more effectively. Anticipate resistance before it develops. Manage the disease's evolution rather than simply trying to overpower it. The road from here to cure, for more cancers, for more patients, runs through that deeper understanding. It's being built slowly and expensively and with heartbreaking setbacks along the way by researchers who are operating in one of the most complex problem spaces in the history of medicine. The word cure has been said in that consulting room for some diagnoses and some patients with increasing frequency. The aspiration is that the list of diagnoses where it can be said honestly continues to grow. And the reason it hasn't happened faster and the reason it will continue to be difficult even as it improves is the nature of the enemy, which is not an enemy at all, but your own biology, doing what biology does in a way that has gone irreversibly, catastrophically wrong. That's the hardest kind of problem to solve, and the most important